Morphogenesis, a biological process resulting in the organized form of cells and their structures. It controls the spatial distribution and shape of an organism during embryological development. Morphogenesis is achieved through the use of cell-to-cell communications. These communications, including juxtacrine signaling, paracrine signaling, differential cell affinity, differential cell adhesion, extracellular matrices, and morphogen gradients. Juxtacrine signaling is a contact-dependent form of cell-to-cell communication. They communicate through heterophilic and homophilic binding. Heterophilic binding involves binding between two different types of receptors, and homophilic binding involves the binding between the same type of receptors. They transmit signals directly through this type of binding into the neighboring cell. An example of juxtacrine signaling is the Notch pathway and the interaction between the Notch receptor and the delta ligand. Paracrine signaling involves the secretion of proteins into the extracellular matrix. Signaling proteins are also known as ligands. These proteins are then identified by specific receptors in the cell. Once these signaling proteins attach to the receptor, they are either taken into the cytosol or nucleus of the cell. An example can be seen in neutrophils that release chemokines to attract other cells through chemotaxis. In 1955, Towns and Holt-Frater found that cells present in the embryo showed some differential cell affinity that exhibit selective affinity. This was seen in the selective affinity between the endoderm, ectoderm, and mesoderm. The experiment consisted of mixing the cells, and the results showed that the cells would reaggregate into their respective locations. The endoderm had affinity for the inner layer of the cell and would reaggregate into the internal layer. The mesoderm had affinity for the middle layer and would reaggregate between the endoderm and ectoderm. The ectoderm was found to have affinity for the outer layer of the cell and would reaggregate into the external layer. The differential adhesion hypothesis model was proposed by Malcolm Steinberg in 1964. He stated that cells follow a pattern of cell sorting based on thermodynamic principles, and that cells will aggregate with the smallest interfacial free energy. Later, modern technology experiments were performed that showed cell types had different surface tension, and the cells with the lowest surface tension were found in the outer portion, and that the cells with the highest surface tension were found in the center. Cadherins are the primary molecules that mediate cell adhesion. They are transmembrane proteins that interact with other cadherins on adjacent cells. Their external domains serve to adhere cells together while also linking and assembling the actin skeleton. They can also initiate and transduce signals that lead to changes in gene expression. These qualities are paramount in morphogenesis. One example of this is the fact that otherwise identical cells with different quantities of cadherins will sort the higher density cadherin cell internally. Another example is how the type of cadherin can affect sorting behavior. If cells expressing equal numbers but different cadherins are mixed, two distinct mounds are formed with one common boundary. The extracellular matrix is the environment in which cell-to-cell interactions occur. This insoluble network of macromolecules secreted by cells provides the scaffolding for cell adhesion, cell migration, and the formation of epithelial sheets and tubes. Extracellular matrices are comprised of the protein collagen, proteoglycans, and a variety of specialized glycoprotein molecules. Proteoglycans are critical in the delivery of paracrine factors to receptors responsible for many functions, including cell morphogenesis. Fibronectin is a very large glycoprotein dimer that organizes the matrix and cells into ordered structures. Its adhesive quality allows for the linking of cells and substrates while also providing a path for cell migration. Lastly, laminin and type IV collagen make up the basal lamina. This type of extracellular matrix acts as an anchor for epithelial cells and plays a role in promoting cell adhesion, growth, shape, and migration, all of which are important for morphogenesis to occur. Morphogens are signaling molecules that form a gradient by moving away from a restricted region of tissue. Cells can have different fates depending on the concentration of morphogens in their vicinity. The French flag model displays this by showing that having a greater diffusion distance can cause cells to differentiate differently.
For the first 15 days, we can see the first heart field cells create a crescent shape in the anterior embryo, with the second heart field cells medial and anterior to the first heart field. On the 21st day, the second heart field cells lie dorsal to the straight heart tube and begin to move into the anterior and posterior ends of the tube to create the right ventricle, the conotrochus, and part of the atria. After 28 days, a right forward looping of the heart tube, cardiac neutral crest cells migrate into the outflow tract from the neutral folds to separate the outflow tract and pattern the bilaterally symmetric atrial arteries. Finally, after 50 days, separation of the ventricles, atria, and atrioventricle valves result in a four-chambered heart, leading to the ventricle, left atrium, right atrium, aortic sac, aorta, pulmonary artery, right subclavian artery, left subclavian artery, right cardioid artery, left cardioid artery, and the ductus aorteus.
The end.